In 1996, two college students were watching boat races along the Columbia River near Kennewick and Washington when they noticed something unusual protruding from the shallow water near the shore. At first, they thought it might be a rock, but upon closer inspection, they realized they were looking at a human skull. They immediately contacted authorities, and within hours, archaeologists arrived at the scene. What they discovered over the next several days would ignite one of the most intense controversies in American archaeology. The remains were remarkably complete, one of the most intact ancient skeletons ever found in North America. Initial examination suggested the individual was male, approximately 40 to 55 years old at death, and based on bone morphology, appeared to have features that didn't quite match modern Native American populations. But the real shock came when radiocarbon dating revealed the skeleton's age. This man had died approximately 9,000 years ago, making him one of the oldest and most complete human skeletons ever discovered in the Americas. Scientists named him Kennewick Man, and for the next 2 decades, he would become the center of a fierce legal and scientific battle. And when DNA technology finally advanced enough to analyze his genetic material, what researchers found would raise entirely new questions about the identity and origins of the first Americans. The controversy surrounding Kennewick Man began almost immediately after his discovery, based on initial skull measurements and facial reconstruction. Some anthropologists claimed he didn't look Native American at all. Instead, they suggested he might resemble Polynesian, Ainu from Japan, or even European populations. This sparked wild speculation in the media. Some fringe theorists used Kennewick Man to promote discredited ideas about ancient European voyages to America, or to question whether Native Americans were truly the first inhabitants of the continent. Native American tribes in the region, including the Umatilla, Yakama, Nez Perce, and Colville, immediately claimed Kennewick Man as an ancestor and demanded his remains be returned for reburial according to traditional practices, as required by the Native American Graves Protection and Repatriation Act. But a group of scientists sued for the right to study the skeleton, arguing it was too old and too scientifically valuable to be reburied without thorough analysis. The legal battle lasted nearly a decade. The case revealed deep tensions between scientific inquiry and indigenous rights, between the desire to understand the past and the need to respect the dead. For years, Kennewick Man's bones sat in museum storage while courts deliberated. Physical anthropologists who were eventually granted access studied every aspect of the skeleton. They documented healed fractures, signs of a hard life, including a stone projectile point embedded in his hip that had healed over years, suggesting he survived a violent encounter. They analyzed his diet through isotope analysis of his bones, revealing he ate primarily marine foods, salmon and other fish from the Columbia River. They created detailed 3D scans of his skull, but the one test that could definitively answer questions about his ancestry, DNA analysis, remained out of reach. The technology simply wasn't good enough yet to extract viable DNA from 9,000-year-old bones. That changed in 2015 when advances in ancient DNA sequencing finally made it possible to extract and analyze genetic material from Kennewick Man's remains. A team led by Eske Willerslev at the University of Copenhagen, working in collaboration with representatives from the Native American tribes, obtained a small sample of bone from the skeleton's hand. Using cutting-edge techniques designed for degraded ancient DNA, they successfully sequenced portions of Kennewick Man's genome. The results, published in 2015 in the journal Nature, were definitive and settled the debate once and for all. Kennewick Man was genetically most closely related to modern Native American tribes from the Columbia River region, particularly the Colville people. He was not Polynesian, not Ainu, not European. He was Native American, and his descendants still live in the same region where he died 9,000 years ago. But while the DNA solved the question of Kennewick Man's identity, it also raised profound new questions about the genetic diversity and population structure of early Americans. The genome revealed something unexpected. While Kennewick Man was definitely ancestral to modern Native Americans, his genetic signature showed he was part of a population that was distinct from some other early American groups. Specifically, his genome differed slightly from the genetic profile of other ancient individuals found in North America, suggesting there was more genetic diversity among early Americans than previously thought. This wasn't evidence of different geographic origins, but rather evidence of population structure, meaning that early Native American populations weren't genetically uniform. Different groups had begun to diverge genetically from each other, developing unique genetic characteristics, even though they all ultimately traced back to common ancestors who had migrated from Asia. The DNA also revealed Kennewick man carried genetic variants associated with several physical traits. Analysis of genes related to skin pigmentation suggested he likely had darker skin than modern northern Native Americans, though not as dark as equatorial populations. His hair was probably dark and straight. Eye-color genes indicated brown eyes. Interestingly, he carried genetic variants associated with increased risk of certain health conditions, including genes linked to periodontal disease, which physical examination of his skeleton had already confirmed he suffered from. This demonstrated how genetic and physical evidence could corroborate each other, providing a more complete picture of an individual's life and health. Perhaps most intriguingly, Kennewick man's genome contained traces of ancient admixture, genetic evidence of interbreeding between different human populations that occurred deep in the past. Specifically, scientists detected small amounts of genetic material from Denisovans, an extinct group of archaic humans known primarily from a few fragmentary fossils found in Siberia. All Native Americans carry some Denisovan DNA, inherited from ancient interbreeding events that occurred in Asia before their ancestors crossed into the Americas. But the specific pattern and amount of Denisovan ancestry in Kennewick man's genome provided clues about when and where this interbreeding occurred, helping to map out the complex journey of human populations from Asia into the Americas. The genome also shed light on the relationship between ancient and modern Native American populations. By comparing Kennewick man's DNA with genomes from hundreds of modern Native American individuals, researchers could trace lines of ancestry and estimate when populations diverged. The data showed that Kennewick man's lineage split from the ancestors of some South American indigenous peoples approximately 14,000 to 15,000 years ago, shortly after humans first entered the Americas. This suggested that by 9,000 years ago, when Kennewick man lived, the genetic structure of Native American populations was already becoming regionally differentiated. People living along the Columbia River had distinct genetic characteristics from those living in South America, even though they shared common origins just a few thousand years earlier. This finding challenged the idea that ancient Americans were a genetically homogeneous population that only diversified in recent millennia. Instead, it suggested that genetic differentiation began very early, possibly due to geographic separation, adaptation to different environments, or simply genetic drift as small founding populations expanded and spread across vast territories. The Columbia River region, where Kennewick man lived, was a particularly rich environment with abundant salmon runs that supported relatively large and stable populations. These populations could have remained somewhat isolated from groups in other regions, leading to the development of unique genetic signatures. The Kennewick man genome also raised questions about what happened to his lineage. While he is ancestral to modern Columbia River tribes, the genetic connection isn't direct and continuous. There's evidence of population turnover and replacement in the region over thousands of years. Some genetic lineages present in Kennewick man are rare or absent in modern populations, suggesting his direct descendants either died out or were assimilated into later arriving groups. This pattern of population replacement and mixing appears to have been common throughout the Americas during the Holocene, the last 10,000 years. The genetic landscape of Native America wasn't static. Populations moved, mixed, replaced each other, and evolved, creating the incredible diversity of indigenous peoples that existed at the time of European contact. One of the most significant aspects of the Kennewick man DNA study was its collaborative nature. After years of legal conflict, scientists and tribal representatives came together to conduct the research in a way that respected both scientific goals and indigenous concerns. The tribes agreed to allow DNA testing, scientists agreed to limit the amount of destructive sampling, and all parties committed to reburying the remains once the analysis was complete. In 2017, after the DNA results were published and additional studies completed, Kennewick man, now referred to by his tribal name, the Ancient One, was reburied in a secret location along the Columbia River in a ceremony attended by representatives from the 5 tribes who claimed him as ancestor. It was a powerful moment of reconciliation and respect. The story of Kennewick Man demonstrates how DNA evidence can resolve long-standing questions while simultaneously opening new avenues of inquiry. His genome confirmed he was Native American, settling the identity question. But it also revealed unexpected genetic diversity among early Americans, evidence of population structure, regional differentiation, and complex patterns of ancestry that we're only beginning to understand. Every ancient genome sequenced adds another data point, another piece of the puzzle. And often, as with Kennewick Man, the answers we get prompt us to ask better, more nuanced questions about who the first Americans were, how they lived, and how their descendants became the diverse indigenous peoples that inhabit the Americas today. The unknown DNA isn't unknown because it's from some mysterious lost race or ancient Europeans or Polynesians wandering the Americas. It's unknown because it represents populations and genetic diversity that no longer exist in the same form today. Ancient peoples whose specific lineages have been reshaped by thousands of years of evolution, migration, mixing, and change. Kennewick Man's genome is a snapshot of that ancient genetic landscape, a glimpse into a time when the peopling of the Americas was still unfolding, when populations were still spreading, adapting, and differentiating into the rich tapestry of cultures that would eventually fill two continents. Understanding this ancient genetic diversity has profound implications for how we think about Native American history and identity. It shows that indigenous peoples have deep, complex histories in the Americas that stretch back thousands of years, with as much internal diversity and change as any other region of the world. It challenges simplistic narratives and reminds us that the past is always more complicated than we imagine. And it demonstrates that the best science happens when researchers work respectfully with descendant communities, acknowledging that these ancient remains aren't just data points, but ancestors deserving of dignity and respect. Thanks for watching. If you found this story of ancient DNA and the mysteries of the first Americans fascinating, don't forget to like, subscribe, and share this video. Let us know in the comments what other ancient DNA mysteries you'd like us to explore. Until next time, keep questioning and keep learning.